Is it up? All good?
Looks good. All right, it's my pleasure to introduce our first speaker today, and it's Dr. Taylor Hackman. She's a postdoctoral researcher in Dr. Esteban Soto's Aquatic Animal Health Lab at UC Davis, where she works on a range of bacterial pathogens of fish. When she's not in the lab, she spends most of her time with her favorite terrestrial animal, her dog Willow, who we may hear in the background during the talk.
Yeah, she made her appearance or her presence known last summer. So we'll see if she finds something fun to work at this time too. Yeah, hi everyone. I'm excited to share with you guys some, I think, fun work. I alluded to it last summer, and we've done some more. We've gotten some more progress done on figuring out more about these Streptococcus dysgalactiae infections that are emerging in fish, and the sort of unique genetics behind. So first, I really need to send a big thank you to all the collaborators and contributors for this project. It's been a highly collaborative, ongoing effort. So it's a gram bacterium. It already has an association with causing disease in animals and humans. It's been traditionally classified into two different subspecies on the basis of hemolytic profile and source of isolation, though those have held up relatively well with the advent of molecular genomics. So the first subspecies, S. dysgalactiae, subspecies dysgalactiae, is generally alpha or gamma or non-hemolytic on blood agar, and it's typically associated with infections in cows primarily, but also other animals. In contrast, the second subspecies, S. dysgalactiae, subspecies equisilamus, is beta hemolytic, most often on blood agar, and comes primarily from human clinical cases. Though, of course, there's a few horse isolates in there from which it was first described. Exceptions, however, to these denotations, this division, have been reported with increasing frequency, and there's been an increasing rate of both human and animal cases caused by both subspecies in the recent decades. Okay, so we've already started to sort of call into question this two subspecies delineation. Streptococcosis caused by S. dysgalactiae is also an emerging disease. So our versions of Piscine streptococcosis, right, are going to be S. aeneae and then S. agalactiae, and particularly in tilapia. But we're getting increasing reports of these atypical causes, including group C Streptococcus dysgalactiae infections. It was first reported in 2002 and has since been popping up in both fresh and marine water, cultured and wild species, primarily in Asia and then in the Americas. These fish-associated isolates were attributed to Strep dysgalactiae, subspecies dysgalactiae, and that was mostly on the basis of their hemolytic pattern. So most of them were alpha hemolytic, at least at 24 hours, or single gene sequencing of either the 16S rRNA gene or the SOD-A gene, which is what is shown in this phylogenetic tree. But as you can see here, it's been long appreciated that the fish-associated isolates form this sort of distinct clade separate from the mammalian. So we've been observing these trends, but grouping them with SDSD, subspecies dysgalactiae. Recent work, however, that's incorporated more of a whole genome approach has suggested that they should instead be grouped with subspecies equisilamus, or even within their own subspecies. So Porcelato et al. Did a really lovely paper that wasn't focused on fish isolates, but included some. And in that, he hinted that those isolates maybe should be considered their own subspecies, as well as some of the other SDSE animals host-specific clades. And then our recent publication that will hopefully be coming out soon with Dr. Shoemaker, we also are isolates from Nile tilapia, we're grouping closer to Streptococcus dysgalactiae equisilamus by multi-locus sequence analysis. So all of this together led us to want to more systematically and conclusively characterize these fish-associated isolates of Streptococcus dysgalactiae by both genetic and phenotypic analysis. So to do this, we used isolates collected from outbreaks both in a hybrid sturgeon farm in Uruguay and from tilapia in the US. So these were a range of years. The first was from 1998 and the most recent from 2020. And again, from both North and South America and 2 host types. So to first take a look again at the genetic identity of these isolates, we used multi-locus sequence analysis, which many of you have heard me talk about this scheme before. So I developed it for Streptoniae, but it very, very kindly works for other species of Piscine strept as well. So we compared our 18 outbreak isolates with 48 reference S. dysgalactiae genomes of both equisilamus, dysgalactiae, and uncharacterized available on NCBI. And what we found was pretty cool. So there's our big tree on the left, which we can break down a little bit more into a simpler form. So we have our S. dysgalactiae subspecies equisilamus from humans, and that groups separately from those from horses. But it also groups more closely to 2 groups of our fish. Our sturgeon isolates form their own clade, their own distinct clade with each other. And then our fish isolates, both from tilapia and all of the referenced fish strains available on NCBI, group together in a second distinct clade, along with 2 human isolates that are from cases of suspected zoonosis from interacting with fish. So I'm still calling them fish associated because the origin of the isolates was likely from fish. And then down at the bottom, we have our Strep dysgalactiae subspecies, dysgalactiae isolates from cows and humans, right? Because as mentioned, they're popping up in people now too. And so to get an idea of how closely related these groups are in addition to the phylogenetics, yeah, so that's that's the Porcelato paper on suggesting that these host-specific lineages might be their own subspecies. And then I took a look at the average sequence identity between our groups. So these are averages of all of the strains of SD, all of the strains of SDC, and from the fish 1 clade and fish 2. So we can see here that our conventional subspecies are the most distinct from each other. But our fish subspecies aren't closely related to either, right? So the subspecies delineation for a full genome characterization is around 98% identity. So we're falling under that. So it's a good hint that maybe these are neither conventional SDSC nor C. So obviously then we whole genome analysis to get a more comprehensive picture of what these isolates are doing in relationship to each other. So we used a hybrid assembly approach to do both a core genome based phylogeny and a comparison of average nucleotide identity between a representative isolate of the sturgeon clade, a representative isolate that was already available from fish clade 2, and then a number of reference genomes available for the 2 established terrestrial subspecies. So what we found really consists of what we saw for MLSA, which is always a great sign. So in the first group, we have primarily human-associated strains of Streptococcus dysgalactiae subspecies equisilis. Right? So we can see those form a really nice group. And then we have our horse strains as well, still grouping separately. We have our Streptococcus dysgalactiae subspecies dysgalactiae. And then we have two lineages branching off, one for the reference strain from fish clade 2, which contains the tilapia and other fish isolates. And then our sturgeon isolate, representative isolate STC3. When we looked at the ANI calculations for these different isolates, and again, the conventional cutoff for a subspecies delineation is around 98% or less. We can see that those two fish-associated lineages share right under the cutoff with each other. So right now, it looks like they're potentially two different subspecies, but you know, this is only comparing one isolate from each group. So we're gonna roll into further comparisons to make sure to try to get a handle on what the differences are in average between those two clades. But for sure, the human and horse isolates also fall into that range of interest as well, under the 98%. And all of these different groups were definitely under the 98% from each other. So we're at least seeing some very specific groupings from each other, which is cool. And they seem to be a little, in part, host associated. This is also the first genome from that sturgeon isolate. So Hassan and the folks over at Mississippi State did an amazing job getting these genomes together. So the STC3 genome, you can see from the circular comparison graph that it has some really nice unique regions compared to, again, our fish clade 2 isolate from amberjack, as well as our Streptococcus and Epiphylamus ATCC strains. We of course want to know, okay, we've got a genetic difference. What are some of the biologically relevant, perhaps, differences or the phenotypic differences between them? So to start, we saw some really actually very interesting differences in hemolytic profile. So streptococcal strains, part of their characteristics is whether they are hemolytic on different types of blood agar. So on sheep's blood agar, our strains actually showed different hemolytic profiles depending on clade. So the sturgeon isolates were kind of interesting in that all of them except for one isolate, STC2, started out as non or alpha hemolytic at 24 hours and then had very strong beta hemolysis by 48 hours. Whereas our tilapia strains stayed either consistently alpha hemolytic or non-hemolytic over time. Yeah, STC2, I tested it multiple times. It's beta hemolytic from the get-go, more similar to the traditional Equithylamus phenotype. Hemolysis pattern can change based off of oxygen availability. Streptococci often show greater hemolysis in aerobic conditions, and these were tested at aerobic. And then interestingly, in our other work, Dr. Shoemaker observed induction of beta hemolysis in those tilapia strains with exposure to particular antibiotics. So there's an interesting little puzzle here that I'm hoping to learn more about and dig into. But if any of you have come across this before, I'd love to chat. And then we also did some preliminary antimicrobial susceptibility testing. There were some mild differences in the conditions used in terms of aggregate. So we are going to continue to run these to standardize them. But right now, one of the more interesting things that came up that I wanted to share is that it looks like the sturgeon representative isolates were uninhibited by oxytetracycline, which is one of the 3 drugs approved for use in aquaculture. So that's potentially concerning. But again, we'll need to do some further testing to confirm these trends. Oh, right, and it was for oxytetracycline and fluorophenocal that transition from alpha to beta hemolysis was observed for the previously alpha hemolytic strains of tilapia isolates. We also took a look at the fatty acid profiles. So this was Julio and the folks out at USDA. And so it looks, again, some interesting things. That SDC2 isolate that showed the more typical hemolytic pattern for Equisilimus grouped with the human isolates, so our typical isolates of Streptococcus dysgalacticae Equisilimus. So there might be something going on there that we can't see in the MLSA, and it would be interesting to get a look at the genome of that particular isolate to see if we could piece out what's causing that difference in hemolytic and in hemolysis and expression of those hemolytic proteins. But the remainder of the sturgeon isolates had very consistent, pretty similar fatty acid profiles that were distinct from the tilapia isolates. And those tilapia isolates, along with a silver carp isolate collected by the same group, Dr. Hawk, had a little bit more variation in their profiles, but again, tending to group closer to each other. So in order to look at whether that apparent host association for the genetic clades was relevant in terms of virulence, here at UC Davis, Dr. Thunaguin, sorry if I butchered your name, ran an experimental challenge. So she has developed a method for testing streptococcal infections in sturgeon, which are recalcitrant to IP injection. So we took a representative strain from clade 2, a USDA 20A strain from tilapia, and a representative strain from the sturgeon clade, STC1. We stressed the fish out for about 30 seconds twice by netting them and suspending them. And then we used an insular challenge. We did 6 fish per treatment and a CFU of 10 to the 8th. And monitored them for 21 days at 25 degrees Celsius. So we didn't have high levels of mortality, but we did see an interesting pattern in that our SDC1 sturgeon isolate similarly caused the highest level of mortality in sturgeon. So the USDA strain didn't cause any mortality in tilapia, neither did SDC1, so no tilapia mortalities. And then we had limited mortality caused by the tilapia strain injected into sturgeon, but about 50% mortality in with the sturgeon isolate. And our histopath supported some of these lesions that we saw in our fish. So you can see the skin lesions on our sturgeon in the top, and then some lesions in the internal organs as well. So we had hepatocellular evacuation in our fish that survived the challenge. So there's a reduction in the reduction or the absence of hepatocellular evacuation in those survivors, which can be associated with a number of factors, including stress and disease. And also had myonecrosis, inflammation. We lost the overlying epithelium probably due to that injection. And then it's to see the bacteria right in those tissues. So we saw intralesional coccidia, both at the injection site and in other organs. So those were also in the kidney, heart, and liver, often within the vascular spaces. So where we're at now is that those fish isolates are more closely related to SDSE and previous cases, including those historical isolates that were reference strains now in NCBI, were mistakenly identified as SDSD. So they're closer to Equithylamus. But we have two distinct fish-associated clades, and those clades do seem to have some biological relevance. So clade 1 is our sturgeon isolates from Uruguay, and clade 2 is our tilapia and other fish isolates from the US and Asia primarily. We see differences between those clades in hemolytic and fatty acid profiles, as well as potential differences in MIC profiles and potential host virulence associations, but both of those we'd like to explore more. And then there's, do these two clades represent two potential new subspecies of Astiscolactiae? So I'd like to acknowledge all of our collaborators, all of the different institutions. This wouldn't have been possible without them, as well as our funding sources and my lab home at UC Davis, the Aquatic Animal Health Lab. With that, I'll take any questions.
Awesome, great job, Taylor. Feel free to unmute yourself and you can ask the question directly, or you can put it into the chat box. So Taylor, you got a question here in the chat from Dave Groman. He's wondering, did you notice much infiltration of lesions by various leukocytes or not?
So for the histo, trying to remember. So this was done last year. I would like to say yes. I would have to go back through and confirm. There were, let me go back and look at that so real quick. Oop. Yeah, I'm not seeing, especially in the histo I chose to include here. I would have to go back and look at our full histo report. I apologize, I can't remember.
Hey Taylor, I'll chime in. Fantastic talk as always. Really cool stuff. Just out of curiosity, I remember when Dr. Hawk and we were kind of that first isolate from the silver carp, and at that time, there wasn't a whole lot of material in GenBank, so it was matching something out of Japan, I guess. And I guess I'm wondering, is there any associations with like marine versus freshwater fish? I know you said there was host associations, but I wonder if there's some environmental selective process that's driving some of these differences as well. Those are pretty diverse groups of fish you're dealing with. Do you have any? Do you care to comment on that at all?
Yeah, no. So that was interesting. So there really didn't seem to be strong difference between those. At least with the MLSA, I'm planning to expand the whole genome phylogenetics to see if we get any more discrimination between those strains that are resolution, because right now we're seeing really those two clades, and within the clades themselves, regardless. So clade 2 is consisting of both fresh and marine water isolates from fish, from different host fish species, and they're grouping very closely. They shared a high sequence identity between them. So I'm not currently seeing much in the way of environmental differentiation. I was surprised that the sturgeon grouped separately from the rest of the fish. It was a surprise to me, but they definitely do. So far that's the main fact, and it's sturgeon versus every other fish at the moment.
Yeah, no, it's cool stuff. Yeah, we need to set up a working group, kind of collective, make sure we all are, where we're at. And I think we've got most of those genomes to you, so we can, knock that out and finish it up. But yeah, fantastic stuff, Taylor, as always.
Yeah, yeah, that's why I wanted to. This was a bit of a last minute pull together, but I was like, I'm gonna have all the people who are involved.
Yeah, awesome.
To give an update on where
Great
we
job.
are right now.
Great job.
All right, Taylor, thank you very much.
Thanks.